Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gil Eapen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gil@eapen.info. My guest today is Professor Dragana Rogulja, who is an assistant professor at Harvard Medical School. Her interests include sleep, circadian rhythms, and motivation. Welcome, Dragana.
Hi, Gil. Thank you so much.
Yeah, thanks for doing this. So I want to start with one of your papers from 2018 entitled Motivation, Perception, and Chance Converge to Make a Binary Decision, in which you say we reveal a central role for chance neuronal events in the decision of a male flight to court, which can be modeled as a coin flip with odds set by motivational state. That's a really interesting area. So what you're saying here is that the probability of an event happening is clearly driven by motivational state, but it's not solely driven by motivational state. There is a chance that gets superimposed on that, and ultimately that both of these drive an outcome.
Yes. So I guess what we're trying to say is that we and other animals are not really robots. There are chance events involved in all of our decisions. So this study started from the sort of the motivation for the study was our interest in understanding motivation. So what is it that motivates animals to do something or not? Because you're always surrounded by sensory cues. For example, you can see a mate or potential mate, another animal that you are interested in courting or not. You can see food that you decide to eat or not. You can see a bed, and not every time you see a bed you decide to sleep. So a lot of variables go into the decision of animals to do something or not. And so we wanted to understand basically how animals make decisions to start doing something. And so the first thing that we found was that in male flies, so we use male flies as a model system to understand motivation, and specifically we use their sexual behavior. And we found that male flies are very interested in courting female flies and mating with them at first, but then after mating several times, their propensity to go after females, their interest declines as they use up their reserves of sperm and seminal fluid. They lose their interest in female. And then we explain how this internal state, whether they have mated or not, influences their decision to then pursue a female. And so this is the motivational state, essentially, when you go from not engaging in mating, you have, or before the male, or a naive male, let's say, before he engages in mating in a female, he has very high mating drive. And then as he engages in several bouts of mating, he loses the interest in female. And so we explain how that internal state biases the propensity of male to court or not. And we explain how dopamine specifically biases the chance of male mating or not. But it is not a completely deterministic thing. So it's not that every time a male that is naive, that has not mated, that every time he approaches a female, he will decide to mate with her. It's also not that every time a male that is satiated, even though their propensity to court females is diminished, it is not that male, that there's no chance that an animal will go after females. So we kind of explain mechanistically what it is that the dopamine does to kind of bias the chance of this happening or not.
Yeah, so you say dopamine signal desensitizes one of the command neurons.
Right.
So it is. So the idea that you have some sort of a random probability superimposed on that motivational state, would it have had some selection advantages? So I guess two questions. Mechanistically, how does that randomness get incorporated?
So the male, when he touches the female or taps a female, he gathers sensory information from her. And after each tap, he makes the decision whether to court or not. And in a high motivational state, the chance that he will court is higher than if he is in a low motivational state. And every time he touches the female, he gets both excitatory and inhibitory inputs onto what we call command neurons, courtship command neurons, which drive the actual courtship behavior itself. And so you can imagine that if you get both excitation, something driving you forward, and inhibition, something sort of holding you back, that you could kind of bias, right, the excitation or inhibition. And so towards more excitation or inhibition. And so what dopamine does is that it essentially lowers the probability that these courtship command neurons, which get dopamine inputs, will be, and get these excitatory and inhibitory inputs, will be inhibited once the male, when the male touches the female, so that he has a higher chance of being excited by her. And there's a constant sort of push and pull between excitation and inhibition. So essentially,
is it the quantity? Is it the quantity of dopamine release that ultimately determines what outcome?
Yes. So dopamine, the actual levels of dopamine are really important there. So we found a small set of dopaminergic neurons that are in the brain. So you have these what we call, again, courtship command neurons. So these are neurons that drive sexual behavior, courtship of a male towards female. Okay, so these are neurons that lie upstream of motor actions that the male takes, right, when he courts the female. And we found in 2016, and this is the work, collaborative work with Crickmore Lab, and these experiments were done by Stephen Zhang, a shared student between my lab and the Crickmore Lab. And what Stephen found is, first of all, established this paradigm that males get sexually satiated, that male flies get sexually satiated. And then he found essentially that there is an input that goes from the male's genitals to the brain to regulate the activity of of a small set of dopamine neurons, which then gates the activity of P1 courtship command neurons. So the dopamine neurons are the specific set of dopamine neurons are motivating neurons. So they really, you need dopamine in these neurons acting on P1 So P1 neurons receive dopamine through dopamine receptor. And you need this in order to drive sexual behavior, to drive courtship behavior. And so, yes, dopamine there, and you can actually see in the brain, if you took a brain from a naive male, we mean a male that hasn't mated, and you looked at dopaminergic activity in this small set of neurons, it would be high. But as the male mates several times, 3 to 4 times, you can see the activity of dopaminergic neurons going down, so that these P1 courtship command neurons are basically not pushed into this active state where they would then drive behavior, sexual behavior. And I just want to say that, this is something that has parallels to humans. So a very famous example is from Oliver Sacks, where he, in his book Awakenings, talks about these patients that were essentially catatonic, just not interested in anything around them, almost dead, until L-DOPA came along. And this actually could be given to people, because you can't give people dopamine itself, because of the, uptake, blood-brain barrier permeability, but you can give them L-DOPA, which gets converted into dopamine. And then with that, what he saw is that basically these patients that were catatonic before just sprang back to life. And one of the strongest side effects or correlates of this was their very much increased sex drive.
So clearly, it's a complex electrochemical reaction, ultimately, that ends up in that binary outcome. But like you say, animals are not behaving robotically. There is a significant chance involved. Do you think that idea would have had some selection advantages, and that is why it's continued to be the case?
That things are not completely deterministic, but that there is some probabilistic component. Yeah, I mean, it certainly makes behaviors more interesting.
Yeah.
And having certain flexibility is really important, right, for behaviors. So we see that flexibility is built into systems, into various systems in various ways. And of course, the animal, you can imagine if you're a fly and you're sampling the world around you, that world can also change quickly. If you're, around one animal, that animal could fly away and another animal could come by. And so you're kind of forced to change your behavior sometimes on the fly, so to speak.
Okay. Yeah, so you have a related paper, Recurrent Circuitry sustains Drosophila courtship drive while priming itself to satiety. You say motivations intensify over hours or days, promoting goals that are achieved in minutes or hours, causing satiety that persists for hours or days. Here we develop a Drosophila courtship as a system to study those long time scale motivational dynamics. So this is clearly a very long time scale dynamic. Can we explain this with just the dopamine system, or it's much more complex than that?
So kind of just to break down what we're talking about here is what we noticed, what Stephen noticed, is that after males mate several times, it takes a while for them to recover their sex drive. It takes several days, actually. So the question is, why is that the case? So in that paper, we discovered basically the mechanism through which, first of all, the dopaminergic system is informed about matings, because the question is, so how would you have these dopaminergic neurons in the brain that gate motivational states or to determine motivational states, but how do these dopaminergic neurons in the brain know if the animal has mated or not, right? And so what we found is a set of neurons, again, that I mentioned, that go from the genitals of the male and go project all the way to the brain, where they interact with the dopaminergic system. And so you actually rapidly, after the onset of mating, these neurons, which we call copulation reporting neurons, they talk to the dopaminergic neurons in the brain. And you can rapidly decrease by activating these neurons, these copulation reporting neurons in the brain. Their activity can rapidly diminish males' sex drive. And so what we found is that we discovered this circuit element upstream of dopamine neurons, which is actually, so as I said, these neurons from the genitals go and talk to the dopaminergic system, but indirectly, by first talking to this the circuit element that we call a loop, which consists of several neurons, which can actually kind of hold activity and talk to dopaminergic neurons, and they're a little bit like a battery. And so you're kind of gradually depleting activity in that system and in the dopaminergic system. But so this is a circuitry that you need to drive high motivational state and courtship behavior. But what is interesting in this, so again, when you think about these different time scales, the animals can rapidly satiate their sex drive, and then it takes them a while to recover that sex drive. And so that longer time scale, the recovery of sex drive, we found that is because the motivational circuitry, which sustains high motivational state and courtship behavior, actually drives accumulation of negative or inhibitory elements, which can then push the motivation low. And because some inherent delay steps that are built into the system, for example, having to transcribe certain targets and process them, put them in the membrane, etc., there is a delay that's introduced in the system. So you use electrical activity from the periphery to the dopamine system, sort of to quickly drive some changes in behavior. But then you have this biochemical component that is used to then keep the motivation state low after the male has mated several times. And just to say why would this even be the case is what we found is that after these males mate, say 3 or 4 times, it takes them actually several days to replenish their reserves of sperm and seminal fluid.
And so the system is taking time to come back to where it started.
Exactly.
You say here computational modeling reproduces this behavior in physiological dynamics, generating predictions that we validate experimentally. So we could there is sufficient amount of predictability how long it takes for this whole whole sequence of activities to take place.
Yes. So you're asking how is it that the computational model can, or you're just remarking on how well it aligns with the experimental sort of observation.
How will it so is it, I was wondering, there should be a lot of individual variations, I would imagine, right? But that is not the case.
Well, there are individual variations, I mean, to some extent. And you mean individual variations between animals or between
animals? I was specifically thinking about Freud. Yeah.
So we do see, there is a certain variability in the system. Like I said, some males will get satiated after 3 matings. Some will get satiated after 4 matings. But in general, there's not, as much variability as you might predict, in terms of behavior. And again, what is really interesting is that these animals, their motivation is driven by their reproductive status. So they tune essentially their sex drive to their reproductive potency. So as with each mating that they engage in, they deplete some of their reserves of sperm and seminal fluid. The mating drive is going down. But also what's interesting is that they don't directly measure the volume of fluids they lost. We thought at some point maybe that would be the case. Maybe you sense the stretch of the ejaculatory bulb or something like that. That's not the case. They actually count the number of matings through these copulation, the activity of these copulation reporting neurons. Essentially, because with every mating, you deplete some of the sperm and seminal fluid. So you use the number of matings essentially as a proxy to estimate how much fluid you have left.
Obviously, this is all instinctual, right? And it's not like
It's
the fly
not
is
like
thinking
sitting
about
there and counting, but somehow it knows. Exactly.
Yeah. I want to touch on another paper. I know that you are not an author on this, Dragana, but it's sort of related and interesting. So, calmodulin-dependent protein kinase 2 measures the passage of time to coordinate behavior and motivational state. You say electrical events in neurons occur on the order of milliseconds, but the brain can process and reproduce intervals millions of times longer. And we present what we believe to be the first neuronal mechanism for timing intervals longer than a few seconds. This is one of your colleagues, right?
So I'm on the paper, but I'm not the corresponding author. So this is work from the Crickmore lab, and his student Stephen Thornquist was the first author on this paper. Okay, so we talked so far about courtship behavior, right? So the males, when they're pursuing the females, they have this courtship ritual where they sing to them, extend their wing, vibrate the wing, sing to them, tap them, lick them, etc. And then the female decides whether she wants to mate or not. She pauses, and they start mating. Once the mating starts, then there's a remarkably fixed duration of mating, about 23 minutes. And so during this time, the animals mate, and then the male dismounts, gets off, and they go their separate ways. So one question is, what determines the duration of that interval of mating? And so what the Crickmore lab, Stephen Thornquist, what they found is that within a very small set of neurons that determines the duration of copulation, there is a biochemical timer, CAM kinase 2, which actually determines the duration of a very critical event within that 23-minute timer. So this is what I mean. If you measured how long flies mate, they mate for about 23 minutes. But what the Crickmore lab found is that within the first about 6 to 7 minutes, or let's say for the first 5 to 6 minutes, the male will not give up the mating pretty much no matter what you do to him. So you could threaten his life with, say, high intensity heat that will certainly kill him. He will not break up the mating. And then after about 6 to 7 minutes, even with the very mild stimulus, mild heat, he will dismount. He will give up the mating. So essentially, he goes from a very high motivational state, high propensity of sustaining the mating, to a low motivational state. And what happens is that about 6 to 7 minutes is when he transfers his sperm to the female. So they continue mating for another, after those first 6 minutes, 7 minutes, let's say, they will mate for another 15, 16, 17 minutes. Okay. Yeah. But those first 6, 7 minutes until the sperm is transferred are really critical. Okay. And the male is in this high motivational state. So what determines those first 6, 7 minutes? That's what this CAMK2 timer does. So essentially, you get activation of CAMK2, which is a calcium dependent kinase, and then this enzyme, it autophosphorylates, it becomes active, and it sustains its activity for about 6 minutes. And having high CAMK2 activity prevents these neurons from going off, essentially from the male, from ejaculating and from switching into that low motivation state. So you need to sustain essentially a high level of motivation for those 6 minutes until sperm is transferred, because that is, let's say, the most important event that happens during mating is, that fertilization. So CAMK2 essentially keeps these neurons from going off for a certain amount of time. And it does that through the dynamics of its phosphorylation and decay of that activity.
So just the presence of that essentially provides a higher level of inhibition.
It provides basically what it does is that it prevents, it keeps the male in this high motivation state. It prevents the sort of distracting or threatening inputs from, breaking up the mating, essentially.
Right. Yeah, and again, one could speculate some selection advantages here. Because as you say, the first 5, 6, 7 minutes are the most important ones. And so it seemed to have adapted to giving high levels of importance there, and then it progressively declines.
Yes, absolutely. Yeah. And what's really interesting about this is that a lot of our behaviors, if not most of our behaviors, occur on these kinds of time scales that are on the order of minutes to hours. But there's really very little, almost nothing is known about the timing of those events. So that is, those time scales are referred to as interval time scales. So, neuronal activity occurs really quickly. It happens on much shorter time scales. And then the timing system that we know the most about is the circadian system, which operates on much longer time scales, so 24 hours or so. But really nothing is known about timing of events that are in between. And so I would say that this is like really probably the first instance of a well-described interval timer. And this molecule CAMK2 is used in many different contexts, and it's one of the oldest sort of molecules that we know has operated in living systems.
Mm-hmm. We'll take a quick break, Dragana. When we come back, we'll talk about your very interesting work in sleep loss.
Okay.
And death.
Sounds good.
Okay, talk to you soon. Thanks. This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com. So we are back. Dragana, you have done a lot of very interesting work more recently on sleep loss. There is a paper that just came out, Sleep Loss Can Cause Death Through Accumulation of Reactive Oxygen Species, ROS, in the Gut. You say the view that sleep is essential for survival is supported by the ubiquity of this behavior, the apparent existence of sleep-like states in the earliest animals, and the fact that severe sleep loss can be lethal. But the cause of this lethality is unknown. So are we sure all animals sleep?
No, of course we're not sure of that because, we haven't looked at all animals. But, as everywhere we look, we do see this kind of behavior. And, you can split hairs and say, well, how do we know, even if an animal is basically not moving and not responding to stimuli during a time of this time of disconnect that's like sleep? Well, maybe it's not sleeping until unless we have electrical activity like EEG report from that. But I don't think that is a really good argument. I think that some animals that are simpler than us, their EEGs, for example, would not look the same as us. But the fact is that they do engage in this kind of sensory disconnect state where they don't move a lot. And so that's seen all over the animal kingdom in very simple animals. So you're an animal, you have nervous system, you usually have some form of sleep-like behavior.
Then you have a nervous system. So in terms of single cell organisms, do they have anything similar?
I mean, they have rhythms, yes. So, that there are rhythms of sort of activity, like higher metabolic activity, lower metabolic activity, even in single cells. You have periodic events that could be seen as sort of resting states.
Okay.
But, the way that we think about sleep, we think of that as in animals, essentially.
And water animals like fish and bigger ones like whale and all of that, they all have some sort of sleep.
Yeah,
situation.
they sleep, and even animals like jellyfish have been shown to sleep.
And how about plants?
Flies, yeah, they sleep, and their sleep is very similar to our sleep. So they sleep for many hours a day, and the genes that have been discovered that regulate sleep are really similar between flies and mammals. And the circadian clock, which is sort of an important driver of sleep, was really first described in the fly. And so they're a very good model in which to study sleep for various reasons. Their lifespan is shorter than that of many mammals. So you can do studies like what we were interested in, which is to really look at the effect of sleep loss on the length of life or on survival.
Yeah, so I was just trying to get there appears to be sort of a reason from physics for sleeping. Let me just speculate. Correct me if you think this doesn't make sense. That if you think a biological system as a machine, it's accumulating waste, and sleep is really sort of a cleaning time for the system to get rid of the excess waste while it was operating. Is that fair?
So there's some even kind of direct, claims of actual toxin clearance from the brain, let's say during sleep. And I'm sure that is one of the functions of sleep. The thing that we think about is the fact that animals, again, that are simpler than we are, and that don't even have brains but have very simple nervous systems, they do sleep. And so there's a possibility that there's sort of clearance, even physical clearance of certain substances, even from individual cells or from tissues, but we don't yet know what that is. What we were really interested in understanding is sort of like what the question that we really asked is what is the most basic or requirement for sleep in terms of survival. So why is it that lack of sleep will kill you? And that doesn't necessarily explain the reason for sleep, right? It could be that when you don't sleep, just some adverse events occur that can kill you. It doesn't mean that that's the reason why you sleep. But it solves definitely that really big mystery, which is that it has been observed in various model systems that if you prevent sleep, this causes death. I mean, there just hasn't been, convincing demonstration that you could prevent animals from sleeping and that they could survive without it. So this is what we found too.
Yeah, so you have two, at least two, possibly more experiments, right? So one in flies, one in mice, going through a process of so you have a control and you have a model that is deprived of sleep for a period of time. How many days? So I guess could you talk about the fly experiment first and what you found?
So basically, again, the motivation for us was to understand what lack of sleep does. And let me just say that traditionally, kind of how sleep is usually talked about is in terms of brain and the nervous system, right? So this is for good reasons, because you need the nervous system to go to sleep. And of course, we dream during that time. And we know that if we skip even one night of sleep, we don't feel good the next day. It's hard to think, to remember things, etc. But again, animals that are much simpler than us, they don't even have brains, that have very simple nervous systems. They sleep, and it seemed to us, that not all function of sleep could be explained by these cognitive functions, right? So we didn't just appear with these wonderfully complicated brains, right? So if simple animals, and we have evidence that sleep emerged in earliest animals, and if this is true, then it probably serves some basic function. So our idea was, we really wanted to understand then, what is it that lack of sleep does to the body, or does to the animal. And let me just say that clinical evidence shows that the lack of sleep can lead to various diseases, which is another indication that it cannot really be all about the brain. And so we didn't want to be married to this idea of sleep equals brain. And we thought that, okay, if something goes wrong really with animals and they die, then there should be some signature of impaired cell function or cell death somewhere in the body. So we were not restricted to the brain. And essentially the idea was to deprive animals of sleep and try to do it with different methods of suppressing sleep, of sleep deprivation.
Yeah.
And then look all over the bodies for signs of damage. And so this is the work carried out primarily by postdocs in my lab, Alex Vaccaro and Josef Kaplan-Dor. And what they did is they prevented flies from sleeping. We then saw that this indeed decreases their lifespan severely.
Yeah.
Their lifespan in half. Let's say if you suppress 90% of sleep, or if you prevent all sleep, cuts it down in a quarter. And preceding this increase in mortality, they found that there's a really dramatic increase in reactive oxygen species, and we can talk about what these are, but these molecules, reactive oxygen species, in the gut specifically of flies. And so this accumulation preceded death in flies, regardless of the method with which we suppressed sleep.
So what are some examples of ROS?
Examples of ROS?
Yeah.
So hydrogen peroxide, H2O2, is an example of ROS. That's a relatively mild ROS, or superoxide is another one. So what ROS are the ROS stands for reactive oxygen species.
Yeah.
So these are derivatives of molecular oxygen. So oxygen is a very reactive element.
Right.
So it's a very chemically reactive element. It loves electrons, and that's why we use it in a lot of reactions. Most notably, you use it in the mitochondria, in the electron transport chain, where it's essentially a sink for electrons. You electrically power production of ATP for your sort of energy, cellular energy currency in the mitochondria. You electrically power this, and then what you do with those electrons at the end? Oxygen takes them up because it really loves electrons. But in the process of doing that, it gets converted into an even more reactive molecule, reactive oxygen species, which then needs to be quickly neutralized in the cell. But ROS are made in other processes. They're not just made in the mitochondria. They're made, for example, in the gut in particular, they're used for normal physiological processes. So they're made, for example, to control the levels of bacteria in the gut, and also to control turnover of cells in the gut, which actually do need to be turned over every few days.
Oxidative stress, I know that it has been implicated in many different things, including Alzheimer's disease, oxidative stress in the brain. This is oxidative stress in the gut. And you are drawing a specific connection between lack of sleep and the production of ROS, and then consequent oxidative stress. And the implication is that severe oxidative stress, prolonged oxidative stress, leads to death, at least in the fly experiment.
Yeah. So in the gut, this accumulation of ROS in the gut was followed by oxidative stress, like you said. And basically, what oxidative stress is it's oxidation of cellular macromolecules. And what oxidation is, it's essentially loss of electrons. So you can imagine these reactive oxygen species, the reason why they're so incredibly reactive, the free radical forms of ROS are the most active ones. And what they have is in their outer most orbital, or the valence orbital, where electrons that engage in chemical reactions are, they have unpaired electrons. So electrons are stable if they're paired, if you have pairs of electrons. If you have unpaired electrons, they're very, it makes the element unstable. And so then they try to steal electrons from other molecules, like, cellular macromolecules like DNA or proteins or lipids, in order to stabilize themselves. And in the process of doing that, they destabilize the target molecules, and that's oxidation. And essentially, it's like rusting of metal, or the like apple turning brown when you cut it. That's oxidation. And so you essentially destroy cellular macromolecules, which can lead to death. And so what we found is that actually was proposed many years ago, and by many people, it has been put forward, the idea that sleep loss leads to oxidative stress, that this could be the consequence, kind of a major consequence of sleep loss. And that this was never experimentally shown that that's actually what really causes the demise of the animals. But also it was claimed that this happens in the brain, or it was thought that this happens in the brain. And we could really see excessive oxidation, at least with the methods that we use, where we saw it very easily in the gut. We could not see that in the brain. And in fact, different studies have shown that the level of antioxidant or protective anti-ROS sort of enzymes goes up in the nervous system during sleep loss. And actually, this makes sense in a way that, in a sense that neurons cannot be replaced the way that cells in the gut can. And probably it's really, really important to protect neurons from oxidation. We actually now we think we know the mechanism that leads to ROS production in the gut, but we're still not sure of the details. But what we do know is that if we prevent ROS accumulation in the gut of flies, either by giving them antioxidant compounds orally, and so what some of these antioxidant compounds are, essentially it's like they have a bag of extra electrons that they can give to ROS, to quench their thirst for electrons, so that they don't have to attack, cellular molecules. And so if you do that, you prevent or you neutralize ROS, and animals can survive, have fully normal lifespans without sleep. Or if we genetically target expression of antioxidant enzymes specifically to the gut, we can do the same. So this is in flies, right? And so in flies, we use 3 different methods of suppressing sleep, and we saw ROS. And then what we wanted to, so that was really important for us, because we didn't want, whatever we see to be constrained by any methodological specifics. And then we wanted to know whether this is a conserved phenomenon.
So excess ROS, the excess ROS, very simplistically, Dragana, relates to cellular damage and ultimately death.
Yes, excess ROS. Yeah, and that is really the key. So before we started this study, I didn't really know much about ROS, and I thought of them as bad molecules, right? Because you kind of you hear free radicals, oxidants, antioxidants, etc. But it turns out that they're actually really important signaling molecules at low levels. Okay, so they're important. They do important things in the cell, and you have in place these various actually antioxidant systems that prevent ROS levels from increasing to abnormally high or too high levels. And then if this is disrupted is when problems arise, when molecules in the cell get damaged, and when cells can die. Essentially, you induce cell death.
So I mean, the numbers are quite striking, right? So 100% sleep loss almost goes to about a quarter of the lifespan for the fly. And you found similar numbers in mice models too?
So what we found, so we only did sleep deprivation for 5 days in the mouse.
Yeah.
So
Yeah.
that's what we were allowed to do, essentially what our protocol allowed us to do. And so we didn't deprive them until that. Okay, we just deprived them for 5 days. But that was plenty enough to see ROS accumulation in the gut. So what we did, again, just like what we did in flies, is we deprived mice of sleep, and then we dissected their various organs, and we looked at the level of oxidation or reactive oxygen species. And what we saw again is that there was a huge increase in the small intestine and in the large intestine to a lesser degree. It was in both of those tissues, in small and the large intestine being also known as colon. But it was particularly obvious in the small intestine. And we could not see, again, anything similar in any of the other organs, at least at that time. So after 2 days of sleep loss, you see reactive oxygen species going up. And after 5 days, you see that there is actually oxidation, or the consequence of ROS, or the damage of macromolecules in the gut.
Do we have any human data, Dragana, on this dimension?
So what I can say is that, not exactly like this. So we couldn't study humans yet. We're trying to come up with some ways to indirectly measure level of oxidation, to have some reporters of oxidation. It would be great if we could have something in, like, some fecal markers, right? But what we know is that people which have inflammatory bowel diseases, they actually tend to have inflammation bouts if they don't sleep. If they don't sleep. So, and also there is actually a quite good correlation between disturbed sleep and increased incidence of colorectal cancers.
So this is specifically sleep loss. What happens if you just disrupt the rhythm?
Circadian rhythm, or the rhythm of sleep.
Right.
So you mean, like, if you allow animals to sleep like normal amount, but not at the right time, or something like that?
Exactly.
Yeah.
Exactly.
So we haven't done that. It's something we want to do. You could also ask what happens if you change the quality of sleep, right? If you allow them to sleep the same amount of time, but then they don't sleep as deeply, or something like that. We haven't done that yet. But I'll tell you that there is evidence, again, from humans, that disruption of circadian rhythms, or even timing of sleep, has bad health consequences, such as increasing the chance of colorectal cancer and breast cancer, for example. Because people, for example, they work in shift work, right? They do studies on shift workers, and they see this this problem. So often, people that work in shifts, they probably tend to also not sleep the same amount. I'm not sure, how well that is controlled, but there is evidence that even sleeping at the wrong time is not going to give you the same protective benefit as sleeping, at night.
So you have some evidence that this could be reversed with antioxidants, right? So I'm just speculating here. So I know vitamin C is an antioxidant. So people who are either have disrupted sleep rhythms, or for some other reason, would it be more preferable for them to have antioxidants?
So that is a really good question, and I would say that yes, under the right circumstances. So what I mean by that is that, as we alluded to earlier, antioxidants, ROS, are not all bad. They're actually protective molecules in at certain concentrations. And we actually, there's a possibility, some of our data suggests that perhaps the initial increase in ROS that is seen with sleep loss might be for protective purposes. And because in this case, it's sustained sleep suppression, that you go from beneficial to harmful regimen. So what I mean is that what this suggests then is that, yes, antioxidants would be beneficial, but you want to know when exactly to take them. And that's why we would like to have a system where we can actually monitor directly ROS levels, and when they're going up at the right time, you know, would probably benefit from antioxidants.
So you need to know when and how much.
Exactly.
So if at all
Exactly.
it has to be some sort of sustainable release.
Exactly. And this actually,
release.
you know, explains probably a lot of the literature, people working on antioxidant systems. It's really interesting because there's a lot of contradicting data. You know, in some study, antioxidants are shown to be extremely valuable, and in other studies, people see no effect. So what we conclude is,
but it could also have bad
have
effects,
bad effects,
right? That's
exactly.
something that,
and we actually saw that in certain cases, if we give, so for example, it mattered, how severe the deprivation is. So when do you want to start giving antioxidants? Because in some cases, if the deprivation is mild and you start giving a lot of antioxidants and you start giving them early, that could actually have a bad effect. Okay, so some of these ROS might be made for protective reasons at first, and then because we're artificially pushing the system too far, then they accumulate way too high. But the fact is that none of us really know how much how much sleep you're losing and what the damage to your body is. It's also interesting, I would say, that people often say, I'm fine, I need only 4 hours of sleep, I'm okay. But the fact is, what they mean is they feel fine, sort of mentally. Let's say they even feel fine. You don't feel, the need to sleep. But what we show is that those things can be separated. Your sort of feeling of sleepiness or not can be separated from the need of your body for sleep.
Right. Yeah, and like your data shows, I think the system is so finely tuned that it needs some level of ROS for it to function. But if it exceeds a threshold, it has a lot of negative effects. So it is sort of very finely tuned. And so any intervention, especially with antioxidants, have to be titrated to such precision for it to be really
or at
beneficial.
least to a certain approximation in a certain range. So this is something that we would really love to do. What we would love to develop for the mammalian system is the ability to monitor ROS levels in the gut, and then based on that have develop therapies, interventions, which I think is possible. I mean, honestly, when we saw the results for flies, it took us a while to believe these fly results, because it was crazy. Before we started the study, I don't think we ever would have predicted that we would find what we found, and that we would be able to, once we saw those ROS, it was just so striking. And then the next step was really to show, is this just some crazy good correlation, or is it actually causative? And once we, if it was causative, the prediction would be that neutralizing ROS could extend lifespan. And so intellectually we knew this, but we still couldn't believe it. And then when we saw the results, it was just it was very shocking. So the antioxidants, we screened 53 compounds, and 11 of them were successful at preventing ROS and extending survival. And those compounds that were successful were the ones that were able to neutralize ROS. So not all antioxidants are able to function in the system. And I would say that, coming up with some intervention like this would actually be, I think, enormously valuable, because so many of us are sleep deprived. And right now, the products that exist for people, sleep related things, are things designed to put you to sleep or to help you sleep, right? But not everybody has that problem. It's not like everybody doesn't sleep because they're not able to sleep biologically, right? If you're caring for a young child, for example, or whatever, you're going to be losing sleep no matter what. So it would be really good to have something that you could take and to mitigate some of these bad effects of sleep loss. That's the dream.
Do you have a hypothesis why 11 of those 53, only 11 of those 53 worked? Is there any commonality among the 11?
We didn't really see anything obvious. And we did try for all of these compounds. We tried several different concentrations, but it's possible that for some of these that didn't work, that we just didn't find the right dose, or we should dig into that a little bit more, maybe to think of that. We didn't see anything super obvious as to why some of them work or not. For some experiments with the, enzymes, expression of enzymes, we have an indicator that led us to sort of the conclusion that most likely the levels of ROS are intracellular and not something that's secreted outside of the cell. But I'm not yet sure about the different antioxidants.
Yeah. So in conclusion, Dragana, so where do you want to take this? I was thinking about sleep disruption is a common phenomenon in the modern context, right? Night shifts, pilots, healthcare workers. I mean, you can have a long list of people who have disruption from a natural rhythm of sleep. And so two questions. One is, is it creating problems for them? And number two, if it is there an intervention that might be optimum? Is that a direction you want to take the study?
We want to come up with some ways of mitigating damage in humans. So right now we're doing this in mice, right? So we did it with flies. In mice, we saw that ROS accumulate, but now we're doing a longer deprivation protocol in mice, sort of to see how this influences their health. And then we're going to try to reverse it by antioxidant interventions. But like I said, the dream would be to find some sort of non-invasive way of gathering information from the gut. Okay, so reporters in the gut that would monitor levels of ROS, and then ideally you could couple that with some delivery system that would be triggered at a certain threshold of ROS. So we're thinking about that. And then in terms of so that's kind of a practical thing, but we also want to understand the biology itself. So what is it that triggers ROS production when sleep is low? And then how is it that increased ROS and increased oxidation of the gut leads to death?
Yeah. I wondered, Dragana, uric acid is also a powerful antioxidant. And so if you have excess ROS, I wondered if your uric acid production is lower or anything like that.
I don't know, actually. But I'll say that one of the interesting, sort of interesting antioxidants that worked for us, it worked very well in preventing that in flies was melatonin. And so melatonin in flies is not really known to, it doesn't influence, as far as we know, sleep and circadian rhythms. And this is actually something important that I forgot to say. With those antioxidant interventions, they were capable of suppressing lethality. None of them actually increased sleep. So animals were still sleep deprived, and they were able to. So, but melatonin is interesting because, we usually think of it as related as sort of promoting sleep, but actually its levels are way higher in the gut than they are in the brain. Okay, so there's a lot of melatonin in the gut, and it turns out the melatonin is actually a very potent antioxidant. So it's interesting to think about that, what its function in the gut is. And finally, I want to say that, I think we showed pretty well what happens when animals are prevented from sleeping, and then this can trigger death. But we don't know yet what the normal dynamics of ROS are, if any, in the gut.
And it's, I guess there'll be some variation in it. Obviously it's different for different animals, and it could also be related to any sort of diseases the animal might have as well, right?
You mean as that it could lead to diseases, or that it might be
it might be an after effect of any kind of disease the animal is already suffering from.
Diseases, they sleep and disease, they have it is a sort of a two, it goes both ways. The diseases disturb sleep leads to many diseases, and then that can get worse.
Yeah, I mean, it's an amazing thing, you're losing, no, not losing really, but you're taking off 8 to 10 hours a day. And if that did not have any negative effects, you could have done a lot more
Yes,
work.
but clearly it's something that, I mean, there's a lot of, this is actually kind of useful, is to think, what sleep does, but to think in sort of more specific terms, because, maybe this is why people argue, okay, sleep is required for memory, it's required for this, for that, if we're trying to think of sleep just having one function. But it clearly doesn't, right? If it evolved over, millions of years, of hundreds of millions of years of animal evolution, and we're pretty sure that today it has many functions, such as being required for memory consolidation, etc. But again, there has to be something very basic about it that's tied to its, requirement for survival, and that's really what we're after.
Yeah, I was thinking about that too. Early Homo sapiens, good sleepers, might have been eaten by nocturnal animals. So perhaps there is a selection effect in other direction, but we don't see it.
That's also interesting, and people speculate. Basically, if you were, this is kind of a way to sort of create niches for prey and predator to not deplete the source of food, too fast. So kind of everybody has to sleep at a certain time, So yeah, in a way you're more vulnerable. Right. But actually, from another study that we did in my lab that hasn't been published yet, where we actually found, so this, this study that we discussed, we see this effect in the gut. We have a study now that's not published yet where we show that gut actually controls the depth of sleep. And so if you have a protein-rich meal, you actually can go into deeper sleep due to signaling from the gut to the brain. And so I used to say that, the same thing that you said, and, when you're sleeping, you're vulnerable, etc. But I actually came to think now that you're actually potentially much safer if you're sleeping. If you don't need to go look for food, if you just had a good meal, and you don't have to go looking for food, you're probably better off hiding somewhere and sleeping than you are, running across the field and trying to find some food.
Yeah. So in conclusion, I want to ask one more question, Dragana. Did you find any effects of the microbiome in this process?
No, but we haven't really looked. So the closest we came to that is trying to, with one experiment where we tried to give flies antibiotics and see if this would do anything, and it did not have any rescuing effect. But that's, neither here nor there. I would say we just don't know. We don't know at this point.
Yeah, excellent. Yeah, this is exciting research.
Thank you so much.
And thanks so much. Yeah, for spending time with me on a weekend. Thank you.
Bye.
Sure. Bye.
This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com.